This edition of the Bio Report is brought to you by the California Technology Council, providing discounts on products and services essential to every startup. For more information, visit californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. The use of biomarkers has long held the promise of accelerating drug development and producing safer and more targeted drugs to meet the needs of patients. The explosion of genetic, proteomic, and metabolomic data, as well as the emergence of the human microbiome, is rapidly growing the world of biomarkers. But with that comes challenges for researchers and drug developers about knowing what's available and how well validated these biomarkers may be. We spoke to John Audette, president and co-founder of Amplion, about the company's database, which seeks to make biomarker information accessible to researchers, the role biomarkers can play in improving the drug development process, and how their increased use can bring about a long-anticipated era of precision medicine. John, thanks for joining us.
Thanks for having me, Daniel.
We're going to talk about biomarkers, Amplion's database, and the implications this has for drug development. Let's start with biomarkers themselves. What is meant by a biomarker, and what is the range of substances this may include?
So the fundamental concept of a biomarker has been around for a long time. Biomarker is basically some sort of a biological measurement that you can use to either help diagnose a disease, or to provide some guidance on drug treatment, things of that nature. And so things like blood pressure are actually biomarkers. What's fundamentally changed, though, really within the last 10 years, is that the notion of a biomarker has become much more specific. So as a result of all the advancements that were made during the Human Genome Project in our ability to measure genes and proteins, we now have thousands, tens of thousands of genes and proteins that we know are associated with different types of diseases and other conditions. So a biomarker these days, for the most part, is exactly that. It's some sort of a gene or a protein or maybe even a mutation or a change in one of those very specific molecules that allows you to much more specifically diagnose a disease, decide if you have a poor or a better prognosis for a disease, or to select the right drug. So really the major, characteristic I'd say that's changed over time is specificity. Things have just become much more specific in terms of what you're measuring and what you can learn from that measurement.
As you mentioned, we've had this proliferation not only of genomics and proteomics and metabolomics, but now the microbiome. How good are we at finding new biomarkers, and with that, how good are we at understanding their value?
That's a really good question. I think we're really good at, researchers are very good at identifying associations between genes and proteins and metabolites, as you say, and now elements of the microbiome, and understanding those associations with diseases. Where the challenge comes in is actually putting those discoveries into clinical practice. So one of the things that is kind of characterizes the current state of the art is that there are, literally hundreds and hundreds of new, quote unquote discoveries each year in terms of new biomarkers that have been identified associated with a given disease. This is the next latest and greatest biomarker for diagnosing cancer X. The challenge is, though, that the majority of those discoveries, to be perfectly honest, are not going to end up in clinical practice. And there are a lot of reasons for that. Science is hard. It's hard to reproduce discoveries. Sometimes something's measurable only in a very complex assay that's hard to commercialize. So there are just a lot of different reasons why it can be challenging to actually turn those discoveries into something that we utilize clinically.
Well, let's talk about Amplion. What is Amplion?
So Amplion is a 3.5-year-old company. Started the company back in late 2012 and started building out our team with my first co-founder in early 2013. The goal with Amplion is to put out software products that make it easier for folks to make good decisions about biomarkers and how they can be used in clinical trials and in new products. So our core product is called Biomarker Base. And Biomarker Base is a database that includes thousands of biomarkers that are being developed and utilized clinically. And so one of the things that we're trying to do with Biomarker Base is kind of cut through that noise that I talked about previously, where so many biomarker discoveries ultimately don't end up being useful clinically. And so we raise to our customers all those biomarkers that are being used in at least one clinical trial or being used in at least one diagnostic test. And so those are what we consider the most actionable biomarkers. Once a drug company decides, hey, we're actually going to measure this biomarker in a clinical trial, that's a pretty significant watershed moment in a biomarker's life cycle and shows a lot of confidence in the potential utility of that biomarker. So that's our core product. Our customers are primarily in diagnostic test development, drug development, and they use Biomarker Base to identify the biomarkers that they may want to use in their clinical trials to improve their outcomes, or that for the diagnostics companies, they might want to develop into sort of next generation diagnostic tests.
So how exactly would a researcher or a drug developer use the Biomarker Base? What kind of information does it contain? What would they be seeking to do with it?
Yeah, so a typical use might be for a drug company. They may go in and say, and competitive intelligence is a key use. So they would go in and they would say, hey, for lung cancer, what are all the biomarkers that other companies are using in their clinical trials to identify patients that are suitable for a given drug type? And which of those biomarkers are we not currently using? And so they can actually identify those sort of gaps and then go in and research those biomarkers and say, okay, here, these three actually had significant contributions to positive outcomes in trials. So, these are biomarkers we ought to be using, or these actually didn't end up making any impact. So we're going to strike those from the list. But it makes it very easy for them to stay aware of all the biomarkers being used in different therapeutic areas. And the diagnostics companies, they're really just looking for what are the next either individual biomarkers we should develop into a test. And so they'll kind of track what are the drug companies using to select patients. That's probably going to be a companion diagnostic biomarker down the road if those drug programs are successful. Or increasingly, they're looking for panels. They're looking for what are the biomarkers that are being measured regularly together to diagnose or prognose diseases. And then those are biomarkers that they would consider developing into their own tests.
I imagine the information in the Biomarker Base is drawn from publicly available sources. What does Biomarker Base do that makes it attractive to users? What does it do that other sources of biomarker information doesn't?
Yeah, so that's exactly right. All the sources currently that we draw on are publicly available. And so what we do is make it much easier to utilize those sources for strategic decision making. So for one thing, we aggregate all of the information into one place. So you don't have to search it 3 different FDA websites and an NIH website and, another, PubMed, all these different resources. We aggregate it all into one place. And we also normalize it. So, whereas in all of these resources, maybe you see, a gene is identified 3, 4, 5 different ways, different synonyms, gene symbol, et cetera. We normalize everything down so that you don't have to search for 5, 6 different synonyms to be sure you have total coverage of that area. So that aggregation is really a key part of the value prop. And then we also are able to show trends, right? So that's something you're not able to see at these resources is how are biomarkers increasing or decreasing in use in different applications, either for patient selection, as efficacy endpoints, things of that nature. And so there are competing products out there. The key differentiators for us, I think, are really twofold. They're data quality. We focus very highly on having extremely accurate data in our system. So we actually emphasize that over volume. A lot of some of the other companies that have similar products will really emphasize, just the total volume of information they have in their system. But, there's a lot of false positive in there. There's a lot of stuff that people, frankly, just don't care about. So data quality is key. Also usability. So one thing that's unique about our database is that we utilize a very modern, intuitive web interface for accessing the product, whereas other companies, their interfaces are a bit dated. You've got to go through. We hear regularly from our customers that you may have to check 5 checkboxes to start a search that you can yield a result in our product in just one click. So those are really the key differentiators for us.
One of the issues in using biomarkers, particularly in clinical trials, is validating them. You've alluded to this somewhat, but to what extent are the biomarkers in the database validated, and how is that communication, or how is that communicated to the user, how well validated they may be?
So that's really central to what we're providing here is demonstrating, showing our customers what the validation state for any given biomarker is. It's really key to ultimately getting that biomarker to a point where it's clinically useful. So as I said, the earliest stage biomarkers in our database are those that are used in just 1 or 2 clinical trials. Those are going to be sort of your earliest stage or most emerging biomarkers, and that shows a level of validation, right? If a drug company or some other entity, a research institution is going to say, we're going to measure this in, 400 or 500 patients, even 20, 30 patients, that's a significant cost. So there has to be some validation behind that. And we then also provide all the literature support. So you can actually go in and look at the literature, see how many different publications there are that support the use of a biomarker for a given disease. And so if you see a relatively thin literature out there, it's relatively recent, and it's only being used in a couple trials, that's going to be among your most early stage, least well-validated biomarkers. Then you sort of move up. The next level would be biomarkers used in lab-developed tests, LDTs. So these are biomarkers that don't go through FDA review. They're put out by labs that are offering that test as a service. Then you go to the FDA cleared and approved tests, and then those that are used in drug labels as well. And so those are going to be the highest level of validation. If you go through FDA review at any point, that's going to be certainly the highest level of validation in our system. Now, all that said, one of the things that we're really interested in doing is raising to our customers even earlier stage biomarkers, those that aren't even being used clinically yet. They're preclinical, and maybe they're only described in the literature. So we've developed a very sophisticated data gathering capability that can go out into the literature, find those biomarkers, and now we're building the algorithms that are going to then score them so that you can say, okay, well, there are 20, putative or candidate biomarkers for prostate cancer out there. What are the 3? Here are the 3 that have the greatest likelihood of becoming clinically useful based on certain attributes that we know are similar to biomarkers that are already in clinical use, or attributes that we know confer a greater likelihood of success for that biomarker. So validation is key, and it's really central to kind of what we're trying to provide in terms of understanding levels of validation for biomarkers.
We discussed the proliferation of biological data and biomarkers specifically. What is the impact of that on your database, and how often do you update the information there?
So we update it very regularly. So we used to rely very heavily on manual curation, and most resources like ours are exclusively manually curated. But we're a little bit unique in that we invested very heavily in a data gathering system that uses machine learning and can very rapidly scan, for example, all 200,000 records of clinicaltrials.gov on a nightly basis. And we can then identify new biomarker information in there. And we're constantly training our system to get smarter and smarter about how it identifies biomarkers and whether or not there is actually, the intent to measure it, as we call it. So we can kind of understand the biomarker mentioned in context, and then say, yeah, this is something that our customers are going to care about, and then pull it in. So really, the minimum update frequency that we have right now is every other week. We're probably going to, before the end of the year, be looking at potentially even doing daily updates.
And in terms of the actual growth in the amount of data or biomarkers in the database, how has that grown?
So to give you an example, we started the year with about, I think, 4,000 biomarker indications, as we're calling them, in the database. Those are associations between a specific biomarker and a disease. And obviously, there are going to be, a lot of biomarkers are going to be associated with dozens or even hundreds of diseases. So those specific associations are what we're looking for out there. We started the year, as I said, with, I think, between 4,000 and 5,000, around 4,500 of them. We're currently tracking just over 60,000 biomarker indications for over 4,000 unique genes and proteins. So really, the systems we've developed have expanded our database very, very rapidly.
Drug makers obviously have a big interest in biomarkers because of their potential for cutting the time and cost of drug development. What are the challenges drug developers face in that regard, and how can biomarkers help address the problem?
So biomarkers have really fundamentally changed drug development. They have turned it from a process where you try to get your drug out to the largest possible group of patients. The old paradigm was, we want these mega blockbusters. We're going to give it to millions of people. We all know that, a percentage of those will actually get the benefit. And a lot of, some of the estimates put the average, response rates for drugs at somewhere between 30 and 50%. So, half or more of the people receiving the drugs aren't actually getting any benefit from it. And then a small percentage, hopefully, are actually going to get toxic side effects from it. Well, that paradigm shift is now one where in advance you identify those patients who are going to respond based on some, characteristics that you can identify with biomarkers. And so you end up with smaller cohorts of patients who are receiving the drug. But oh, by the way, during drug development, you have far fewer failures. Your trials take less long because you're able to prove efficacy earlier. They're less costly because you don't have to enroll as many patients in them. So the paradigm shift has gone from, we want to get just a handful of mega blockbusters to dozens maybe of smaller programs that are going to cost less overall and could yield in the end, greater profit for the business overall. We liken it to, they're not swinging for the home runs anymore. It's singles and doubles. It's kind of a Moneyball analogy. And so it's really biomarkers are central to that paradigm shift. As we were early on in the business, it was interesting. Even just, 3.5, 4 years ago, we would talk to drug companies, and we'd go in and we'd find a high degree of variability in those companies in different drug programs in terms of how supportive they were in the use of biomarkers. And not just in oncology, but in cardiovascular and neurology, et cetera. And so, some of these programs would say, we're not interested at all in exploring biomarkers. We know who our patients are. We're just going to stick with this focus. And then other programs would be very much embracing them, even within the same company. But we've also noticed over the years that most companies now have fully accepted the reality of biomarkers, and some are, ahead of the curve in terms of their utilization of them earlier on. So they're already now starting to reap the benefits of biomarker-driven programs. And, they're likely to appreciate some advantages for that.
Even with those successes, I'd argue the move towards precision medicine has been surprisingly slow. I'm wondering, do you think that's because of barriers that are regulatory, that are scientific, that are economic? Why do you think we haven't seen faster progress towards this?
Yeah, I think, and I'd agree with you. It has been slow. I mean, the number of new drugs that we see coming out with biomarkers in their labels is, it's not in the dozens per year, right? It's a handful per year. And the key drivers there, I think, are number one, we're still pretty early on in this movement. Our research shows that it takes an average of 15 years for a biomarker to make it from the point of discovery to the point where it's actually being utilized clinically. And that goes for companion diagnostic biomarkers as well as diagnostic biomarkers. So the companion diagnostic biomarkers that are going to drive precision medicine, it takes a long time for them to get validated and utilized. And so I think that's part of it, right? We're just now starting to appreciate some of the fruits, the early fruits of the Human Genome Project and all that effort that went into developing new sequencing and proteomic technologies, et cetera. And so that's a big part of it. We're just still pretty early. Another part has just been how rapidly pharma and drug development companies have been pushing this, and it's only been, as I said, relatively recently, within I think the last 5 years from our estimates, that there's been a general acceptance that this is the way drug development should be done these days. So that's been a key. Regulatory, I think, has not been such a barrier because FDA on the drug development side has been very proactive in pushing drug companies to utilize biomarkers to the point where it's pretty challenging, I think, to get a submission of any kind for an IND that doesn't include some biomarkers in the program. I think it's just considered pretty much a mandatory aspect of drug development these days. So FDA is actually on that front been pretty proactive. Now on the diagnostic side, regulatory and reimbursement issues have been huge, I think, barriers to advancement. But on the drug development side, I think it's pretty much just been adoption rates by drug companies and their kind of understanding the new economics of this paradigm shift.
Going forward, how do you see biomarkers reshaping drug development?
For drug development, I think we're going to just see more and more use of biomarkers to identify patients for targeted therapies outside of oncology. Oncology has obviously been the huge area where biomarkers have really taken off, and it's because the drugs that are utilized there are targeting specific mutations, and you can identify those mutations as biomarkers. But I think what we're starting to see is proliferation outside of oncology, even in some therapeutic areas, even with some biomarkers that get their start in oncology, they're finding other uses outside of oncology. I think we're also going to see a much greater use of sequencing, next generation sequencing, in identifying patients who are candidates for drugs. So one of the real explosions that's gone on recently is the whole genome sequencing or large scale sequencing of patients, and understanding how phenotypes and genotypes match up in terms of making folks candidates for drugs. So a drug that may have started off for one indication, you could find the parallel biomarker in another disease, and that drug could be a candidate for that individual as well. So I think seeing those crossovers is going to be something we're going to see more and more of as the data that describes those potential drug uses increases. One of the challenges is we just don't have enough data right now, but that's certainly being solved on a number of fronts, both the public and private.
John Audette, president and co-founder of Amplion. John, thanks so much for your time today.
Thanks so much, Daniel. Enjoyed talking to you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.